Today we're going to learn about CODIS and what that is and how that directly relates to what we've learned so far about DNA fingerprinting. So just a quick review. As a reminder, right, we've got DNA in all of our cells, and 99.9% of our DNA is the same as every single other human's DNA, and only 0.1% of our DNA is actually different. Okay. And when you do that by the numbers, 0.1% of our DNA actually ends up being 3 million base pairs. Okay. So out of all the DNA in our bodies, we're really, for wanting to identify someone by their DNA, we're looking at 3 million base pairs. Okay. As a reminder, also back to earlier this semester, we learned about DNA, how it's this double helix, and when it's kind of loose, it's almost like a piece of string or a piece of spaghetti, and then it can coil up, right? Sometimes before our cell divides or what have you, we want it nice and organized, it will coil up. And when the DNA coils up, it becomes a chromosome. So a chromosome is just a coiled piece of DNA, right? We have 23 pairs of chromosomes. So we have 2 chromosome number 1s and 2 chromosome number 2s, and so on. And why do we have 2? Because we get 1 from mom and 1 from dad. Okay. And along our double helix of DNA, we got to think about what is the point of DNA. And the point of DNA is that it gives instructions. Instructions for what? Instructions for how to make things, how to make structures that our body needs. And as it turns out, we have to make proteins. It's all about proteins. So remember, DNA gets transcribed into mRNA, and then that goes to the ribosome, and that gets translated into a protein, right? So those A's, C's, T's, and G's in our DNA is basically just instructions for how to make proteins. Okay. But if we look at our huge long pieces of DNA, if we look at our chromosomes, there are whole chunks of our chromosome that don't actually contain instructions for a protein. It's almost like, imagine if you have a book, right? An instruction manual, and you're like done with step 2 and you turn the page to go to step 3, and there's a page there and it's filled with all kinds of letters, but the letters make no sense. They don't form words, they don't form sentences. There's just a whole bunch of letters there. Okay, so you probably keep flipping in your instruction manual until you get to the next page that actually like says something that you can read. Okay, this is how your DNA is. There will be segments of it that actually make a protein. Those segments are called a gene. And then you will come to segments that there's a whole bunch of letters, but they don't mean anything and they don't make anything. Those segments have been referred to sometimes as junk DNA. They were originally called junk DNA because we literally thought they did nothing. Nowadays, we're learning they might have some other kind of purposes. So sometimes nowadays, scientists prefer to call them non-coding DNA because it might not actually be junk, okay? So we've got these stretches of our DNA that don't make a protein. And so here we go now. Remember that we've got these repeats. We've got these letters that sometimes get repeated in our code. These repeats happen in the junk DNA. It happens in the non-coding part, not for the part that makes a protein. It's in these stretches where it just kind of, there's a whole lot of nonsense there. Okay, that's where our repeats happen. Remember, these repeats are mini satellites. We learned about them already. And the repeats are what we use for the matching purposes. Again, this is part of our junk DNA, part of the 0.1% that makes us different. As it turns out, these repeats can be found in very predictable locations. All humans have genes in certain locations, and they have repeats in certain locations. Okay. And when we're talking about locations on a chromosome, we use a vocabulary word for that. Okay. And that vocabulary word is called loci. Okay. Loci is the plural of locus, just like octopi and octopus, right? Okay. So loci. So basically what loci is, there are very specific points on a chromosome. Okay. So if we look over here with our eye colors here, like this is a chromosome and this is a chromosome, they're a chromosome pair. Let's say this is chromosome number 3 or something. Okay. And you would get 1 from dad, 1 from mom. Like we don't know which is which. It doesn't matter. But let's say this top one's from dad. And so dad gives you the sequence of A's, T's, C's, and G's that code for brown. And mom gives you a sequence from A's, T's, C's, and G's that would code for blue. Right? This person would be heterozygous. Remember those words? Yeah. But the idea here is the brown instructions and the blue instructions are located at the same part on the chromosome. Look, they're like equidistant from the end. They're both on the long arm, the Q arm, right? And so this is called the locus for eye color. This is the gene that's going to make the protein that gives your eyes that color. Okay, so it's not going to be on chromosome 3 on one person and on chromosome 12 on another. No, everybody has their eye color instructions on the same chromosome, but even more specific, on this little same stretch of chromosome. And that's called like a locus or plural loci. Okay, so what scientists have done is they've taken chromosomes, like here's chromosome 7, and they have numbered them. So we know that, like, boom, let's say like right here, they number that 31.1 on chromosome 7. So that if you're talking about a certain gene, you can talk to some other scientists about that specific point on that specific chromosome. It's almost like if you asked me where my house was, and if I just said, oh, it's in Madison, like that's not helpful, right? If I said, oh, well, where's the gene for eye color? On the DNA, that's not helpful. But then if you say, okay, well, where do you live? Oh, I live on the west side. Okay, that's kind of like saying, well, where? Oh, well, it's chromosome 7. Okay, west side, chromosome 7. We're getting more specific, but still not helpful. Well, we're on the west side. Oh, well, I live by Hilldale Mall. Okay, well, now that's more like, oh, well, it's on the P arm of chromosome 7. Oh, okay. Well, now really, where do you live? Oh, I live on 1234 Regent Street, let's say. Oh, well, now I got an address. That's a specific location. So if you say, oh, well, I'm at position 21 and the P arm of chromosome 7, now all of a sudden another scientist is going to go, oh, okay, cool. You're talking about right here. Okay, so a loci is basically kind of like an address of a chromosome. It tells us which chromosome and what part of it. Okay. So as it turns out, there are 13 sites, 13 loci, 13 specific locations on chromosomes that the FBI uses when they're looking to make like a match, when they're trying to match somebody to like a crime or paternity or what have you, okay? And these so these are the 13 sites right here. So like there's TPOX, there's this one, there's that one. Some of them are a combination of letters and numbers, okay? Some of them are just letters. And like you don't need to worry about the naming convention, but like a lot of them start with a D, and then like D3 because it's chromosome 3, D5 is chromosome 5, D7 is chromosome 7, D8 is chromosome 8, and so on. But like you don't need to worry about the naming stuff. But we have these 13 different locations, okay? And these are called CODIS sites. And this word CODIS comes from from combined, D is from DNA, I is from index, and S is for system. Okay. And so the idea here is that the FBI has a whole bunch of stored info of DNA on like people who committed crimes, on DNA that was found in crime scenes. And this is like in an overall database. Okay. They've got 13 sites that they check. All right. And one interesting thing about these sites is because they're non-coding parts of the DNA, you should not be able to identify anyone's hair, eye color, or skin color by these sites, which is really good because, as you may be aware, there are certain races in our society who don't always get treated fairly by the criminal justice system, right? People of color get imprisoned more often and oftentimes falsely imprisoned more often than like white people do. And so using this type of analysis can be helpful because you cannot tell someone's race by looking at these markers. And so hopefully it will take the race card out of this. I'm not saying that the race card is out of criminal justice system, because of course it's not. But at least for the DNA analysis, we shouldn't be able to tell someone's race based off of these specific sites. Okay. So basically, they've got all this database of suspected perpetrators, convicted perpetrators, or just like unidentified DNA at crime scenes. And so if there's been a crime, you can take that DNA analysis and you can run it through the database, all 13 sites, and see, hey, do we get any matches? Oh, wow, interesting. This matches a crime that happened 2 years ago that we never solved. I wouldn't have thought these things were related, but it turns out maybe they are, right? Or maybe it matches the DNA from a site of somebody that was arrested, just recently arrested. And so now all of a sudden you found out, oh, this one person actually did two things. We can convict them for both things or what have you. Okay. So that's the general idea of CODIS and the CODIS sites. So let's look at an example, because I think sometimes seeing an example of how this is used, it's not all that different from what we've seen before. So let's pretend there was a bank robbery, all right? And the bank robber wore gloves because they're professional, but unfortunately they left a hair behind on one of their gloves that they didn't realize. So we take a look at that hair, and they're looking at chromosome number 2. They're looking at the TPOC site in chromosome number 2. And so here, remember how we have 2 chromosomes, one from mom and from dad? So here's the DNA double helix on one of the chromosomes, and here's the DNA double helix on the other. Now, specifically, we're looking at the TPOX region. So this is going to be just the region where our repeats happen. So let's look at these 2 chromosomes. What are the repeats happening here? Like, take a moment, figure it out. I see GAAT, GAAT, or if I look on the other side, it's CTTA, CTTA, right? Same here. Same repeats. GAAT, GAAT, CTTA, CTTA, and so on. So if we look at this chromosome, how many times do we get that repeat? We get it twice, like GAAT, GAAT, or CTTA, CTTA. It gets repeated twice in a row with nothing in between it. How about on this side? It gets repeated 1, 2, 3 times in a row. Okay, so the reason why we get repeats on both sides is, of course, we have complements. So these letters are always going to repeat with their complementary base pairs. So when we do this analysis, we don't need to look at both strands. We just need to look at one strand, because if there's a repeat on the left strand, there's going to be a repeat on the right strand. Okay, repeat's going to be slightly different, but they're both going to be repeats. So from now on, we're just going to be looking at one strand. Okay, so here is our hair in the glove. We're just taking the one strand. So here's our GAAT, GAAT. There it's repeated twice. And here from the other chromosome, it was repeated 3 times. So now we're going to look at 2 different suspects and see if we can figure out who did it. To make this easy, I highlighted this so that the 2 repeats, the double repeat is in yellow and the triple repeat is in green. So I want you to look at suspect 1 and see if you can find any double repeats, either on the first chromosome or the second chromosome. Take a moment, pause the video if you need to. So here is the repeat. Okay, it's repeated twice, and it's not repeated twice over here. Okay, can we find a double repeat on suspect number 2? Again, take a moment, pause the video, see if you can find if there is a double repeat. And it's right there. There's our 2 in a row repeat. All right, let's look for the green now. Let's look for a triple repeat. Let's look for suspect 1. Do we see a triple repeat? And I don't see one. It's not there. Okay, let's look at suspect 2 for a triple repeat. This repeat 3 times in a row. And yep, we totally have one right there. Okay, so which suspect did it? Well, suspect 1 doesn't, although one of the repeat patterns, the other one didn't. And if it's from the same person, they would get both the same chromosome. So we can rule out suspect 1, which means we would be led to believe that it was suspect 2, right? So does this mean that like 100%, yep, suspect 2, throw them in jail, like here we go? Well, not necessarily, okay? Because remember, when we're analyzing stuff, right, we don't just look at one band. We look at, like, in this case, all 4 bands, right? Because, like, this one right here, this bar, for example, look, suspects 2, 3, and 4 had it, but suspect 1 didn't. So we could eliminate suspect 1. And sure, we know suspect 2 did it by looking, but we know suspect 2 did it by looking at multiple repeats, not just one. So one repeat doesn't tell us for sure who did it. It happened to eliminate one suspect, but like, does that mean for sure? Not necessarily. So the odds that one of your repeats at one of your CODIS sites matches someone else is about 1 in 10. So the odds that 2 of yours match would be 10 times 2, or like not 10 times 2, but 10 squared, 10 times 10, 10 times itself 2 times, okay? So that would be 1 in 100. The odds that 3 of your sites would match would be 10 to the 3rd, or 10 cubed, which is 10 times 10 times 10, which would be 1 in 1,000. So if there are 13 CODIS sites, then what are the odds that all 13 would match? 10 times 10 times 10, 13 times is 10 million. So the odds of you matching someone else is 1 in 10 million. 1/10,000,000th of a chance. The odds of you being struck by lightning is 1 in a million. So you are 10 million times more likely to get struck by lightning than you are to have a match with someone else's all 13 CODIS sites. And this is what makes DNA fingerprinting so certain, because the odds are astronomical, okay? And in fact, even since a lot of this educational material has been released, they've added more CODIS sites. So like I'm saying, there's 13, but actually they've added a few more. So then the odds get even bigger or even smaller, you know, that you would have any kind of chance of matching with someone else. Okay, so hopefully after seeing this, you kind of understand the basic idea of a loci on a chromosome. You understand this CODIS database. You get that the FBI use this 13 asterisk, maybe a few more sites when they are matching people. Okay, hopefully you understand walking through, like looking at repeats and identifying the repeats and suspects and using it to figure out like who it might be. Okay, and then the other thing I hope you learned in this is that the little bit of math, figuring out what are the odds of matching at how many different sites. Okay. And tomorrow in class, we're going to be doing some of these practice problems, and you're going to be looking at repeats and analyzing them and trying to figure out like who's the match.